Thank you. It's already introduced me, and my talk is on just like I say, within host problem. You know, most of us concentrate on working on you know, population dynamics. You know, on one compartment of the population to another. But I'm trying to look at within host problem and try to see, can we do something about that instead of waiting for the disease to start before we start talking about the control? Is it possible that we can do something within the host? And it's kind of interrupting the disease establishment in the host first. So if we look at it, our introduction there is talking about, we are trying to bring a narrative in our mathematical modeling approach of epidemiological studies, you know, looking at the human population, and then rather than concentrating on the, you know, controlling the disease within the population, why not also talk about controlling the disease before it penetrates into the cell? Or if it gets into the cell, we stop it from replicating within the cell. Or if it can replicate within the cell, can we stop it from proliferating? Because these are the 3 stages that any disease undergoes, any pathogen undergoes before it can establish a disease in the human system. So I am trying to say, and what I'm trying to do here is to motivate us to try to look at this kind of problem. Not necessarily with looking at it from, okay, you have the disease within you, what will happen, but we want to look at, can we stop it from even getting into your body? And if it gets into the body, can we stop it from subsequent proliferation? So we have started doing some work here, and I have some of my students who are working on the virus, bacteria, and the fungi that we are trying to look at how they enter our body and how they multiply within the body and how they spread within the body. We know that if the disease enters into one cell, definitely when it's entering, it doesn't enter into too many cells. It will enter into 1, 2, or 3 cells, and from there proliferate to other cells. And a reasonable number of the cells must be diseased before the host himself gets sick about it. So that's what I was trying to look at. And I tried to here look at what happens when the disease wants to enter. What happens when the disease has entered? What happens that allows the disease to proliferate within the cell? And so we look at that, and then we try to look at those stages. I took them one after the other. The penetration process, what happens, how does it penetrate? The inversion process, and then the proliferation process. The first one is what we call the penetration. And here we're talking about the disease being able to attach to the cell. And when it has attached itself to the cell, being able to enter into the cell. That attachment and subsequent movement into the cell is what I term penetration process. So there are many ways this can happen. For the virus, we have what we call the receptor-mediator endocytosis. This is when this substance, as a virus or a bacteria, enter inside the cell. It releases the genetic material it contains, which then hijack or alter the reproduction method or other activities. So that is here, the disease, you know, attaches itself to some proteins on the surface, and by that method, it now empties its content into the cell. And when this thing enters into the cell, it disturbs the normal functioning of the cell. And then with that method, over time, they will now develop into a disease within the cell and can now have the processes here, the clustering, the ligand binding, and the vesicle formation. That is, when they get into there, they come for a kind of cyst and detach itself from there and empty the content into the cell. And this now causes a problem. That's why I say these vesicles that are in the cell will eventually uncoat and then release the substance that it was bound with. And then those substances it was bound with outside the cell, or getting into the cell, now will cause problem in the cell. And then we ask ourselves. Then there is another one, what we call the membrane fusion. Remember that when the pathogen is coming, which is the virus or the bacteria or whatever, it has its own membrane. When it comes, it fuses with the membrane of the cell. And through that process, it allows the content to get into the cell. That's why it's talked about the meshing together, thereby allowing the release of viral contents into the target cell. Or this occurs when two separate lipid membranes merge to form a single bilayer. And this, as the name applies, means that it contains a certain specific receptor on the cell surface, and the virus will then release its content into the internal environment of the cell. And has such diseases like herpes, HIV, SARS-CoV-2, they use this method to penetrate into the cell. Then we have the directional injection or penetration method, whereby the virus injects its content into the cell without necessarily entering the cell. To do this, the virus attaches to specific receptors to the host cell surface and pierces the host membrane and introduces its DNA or RNA into the cytoplasm of the cell of the host. In this way, it now empties its content. That's for virus. Then we also have that of the bacteria, because at my interest from now, we are working on 3 types of pathogens: bacteria, virus, and fungi. So if you look at the bacteria also, this one also, we have the adherence to the to the cells or the tissues. This is the actual attachment of the bacteria to the cell membrane of the host cell, and this involves the colonization and specific interaction between bacterial adhesions and host cell receptors. So when the bacteria is forming, it has some adhesive properties, but the host cell which is going to attach to must have the receptors corresponding to that adhesin that is there for the bacteria to attach. So it must have this. Another one is the biofilm formation, which is the aggregation of one or more species of microbes which adhere to the surface and work together to survive. These things, these different bacteria will come together, merge, and then attach themselves to the cell and work together to make sure that they survive. So the adhesion to the surface is first due to an extracellular polymeric substance, which is a sugary secretion made of polysaccharides, proteins, lipids, and DNA that acts as glue to hold the biofilm to the surface. If these bacteria are not there, eventually, even if the bacteria form a colony, they may not be able to attach themselves to the surface of the cell and eventually may not enter into the cell. We also have the adhesin, which is a bacterial protein that mediates bacterial adherence to host cell surface receptors or the extracellular matrix for successful colonization and host cell invasion. If these adhesin are not there, the bacteria eventually may not attach itself to the cell. And the question which we see towards the end of the work is, can this thing be, is there any way we can intercept this to avoid this bacteria attaching itself to the cell? And if it doesn't attach itself to the cell, then eventually the cell will not get diseased. And if it's not diseased, eventually the host will not get diseased. Then we also talked about similar processes of penetration for other agents of disease, such as fungi and vectors. We will not take all of them. Then we now talk about invasion. Invasion now we are talking about when it has gotten, supposing the bacteria or the pathogen of interest has eventually gotten into the cell. What happens next? We talk about replication. If the pathogen inside the cell does not replicate itself, in other words, it does not multiply or does not grow or does not reproduce, then eventually it may not cause any much damage. It may die away there. But remember, as we will see later, that even as the pathogen is fighting to get into the cell is also defending itself against those pathogens. We will see it happen. So if you look at that, then as the pathogen gets entry into the cell, what does it do to the cell? Because it has gotten into the cell, what then happens? T cells like the CD8 T cells pathogen, we now modulate it to make sure that it doesn't recognize it. The next one is that it undergoes antigenic variation to evade the immune system of the cell. It also inhibits the effect of macrophages. We know the function of macrophages when foreign bodies enters into a cell. It produces toxins that damage the cell structure and exoskeletal framework. That's what some of these pathogens does when it enters the cell. And then some pathogen has the capacity to block the process of apoptosis. Apoptosis is self-destruction, where the cell, when it gets certain things that are not normal to itself, and it tries to control it and it couldn't, it will now go for self-destruction, and which we call that. Then, using the available resources in the cell, the pathogen replicates. When it has done all this, blocking the cell from reacting, from destroying it, the next thing you find is that it will try to replicate inside the cell. It tries to reproduce itself into large numbers, which will eventually will now allow it to do more work. Then we now say, while the pathogens can react as above, when it gets into the cell, the whole cell can also react in defense of itself from this pathogen. And what are the ways? One, the whole cell has cell membrane which prevent easy entry of pathogens into the cell. So every cell has the cell membrane, and this cell membrane tries to protect pathogens from entering into the cell. Some of those cells also kill the pathogen as they adhere to their cells of membrane. They release certain substances, certain toxins when foreign bodies. It's just like when we are bitten by ants or something like that. You keep on scratching the point, and as you are scratching the point, you are calling the attention of the natural killers or the macrophages to come up and eat up the pathogen that has entered into your body, or the substance that has entered your body. Similar thing happens in the cell. When these foreign bodies try to attach themselves on the cell also releases some substances that can attack it and destroy it. Also, the cell recognizes the common features of pathogen and so creates tough barrier to their entry, as well as triggering immune response to the presence of the pathogen. Then the whole cell also can initiate signaling cascades such as inflammatory cytotoxins. So this one also to try to cause inflammation at there, and by this inflammation, it tries to block it from having direct entry or indirect destruction into other aspects of the cell. Similarly, the cell can engulf and digest the very pathogen once it is in the vesicle. Remember what we have said, that when it tries to enter, some of them enter in the form of vesicle that bursts eventually into the cell and release the content. But some cells have this capacity that when they notice this vesicle, they will attack it, engulf it, and destroy it. And in that sense, the content will not be released inside the cell. Finally, you can say in a situation where the infection is very severe, the cell, as we have said, can go for a program death called apoptosis. Now, when we have seen this, we now say once the pathogen overpowers the immune defense system of the cell and replicates and establishes itself, the next stage will be to infect other neighboring cells by the process which we call proliferation. So if, however, the whole cell is able to destroy the pathogen, then this disease will be contaminated. But where it cannot, by its own defense mechanism, the next thing the cell is going to do now is to multiply into a large number. And when it does this, the consequence is that it will now either destroy the cell by exocytosis, or it can now release its content. There are many processes which we will see in the proliferation process. And then we say the proliferation here is just the infection of the cell by the same pathogen due to the infection caused by the pathogen in nearby cell. A new cell gets infected because there is a pathogen already in a nearby cell. So if this process is possible for any infected cell to pass the same disease to the next cell, we say the disease is proliferating from one cell to the another. And the process through which this is possible, as we have here, one, direct cell-to-cell spreading of the pathogen, usually common in some viruses and bacteria. And here the pathogen moves directly from infectious cell to another without necessarily entering the extracellular space. What I'm saying here is that the disease does not leave the infected cell and come to the maybe interstitial spaces between the cells, but it will move directly from one cell to another cell. And in this way, the pathogen will be able to avoid infection and attack by the immune system of the cell. So when it moves directly from one cell to another cell without coming out of the cell, it then means that the immune system or the immunity of the, what we call it, the fighting bodies that protect the cells will not even notice externally that there is something because it's moving within the cell from one cell to another. Another one is by lysis, in which case it releases the pathogen, like in the case of viruses. The cell bursts and thus release the pathogen to the extracellular spaces, thereby infecting new cells. When one infected cell don't have enough number of this infected, this pathogen inside it, maybe it then, at a certain population, it will now burst that cell or force the cell to burst it and release its content. Then they will be released to the extracellular space between cells. And in this case, they will now be able, a new pathogen that are released now in a greater quantity will now be able to infect new cells. So unlike in the first case, by this method, the pathogen is exposed to the attack of meat. So in this case, those defense mechanisms by the cells are interplay here, and they can protect the new cell from being infected. Unlike in the first case where it moves directly from inside of one cell to another cell. Another one, some viruses use budding or exocytosis, in which new pathogens will bud off from the membrane of the infected cell, or in the infected cell throws out the replicated pathogen to the external cell, which is what we call the exocytosis, without destroying them, and as such, they can infect new cells. Examples here, we have HIV, we have the HBV, the hepatitis, and many others. Then also, we talk about hijacking the cell transport mechanism. Some pathogens like bacteria and parasites manipulate the host cell machinery, such as the vesicle and the motor proteins, and will be able to travel within and between cells. They hijack the cell transport mechanism. There are transport mechanisms between cells and within cells that allow cells to communicate. And now some of these organisms, when they enter into a cell and replicate inside that cell, they try to hijack this transport mechanism, thereby enabling them to travel within that same process without getting the immune system activated to attack it. Also, we have the viruses formed like the syncytia, which is the mechanism in which the virus will cause neighboring cells to fuse. It will force neighboring cells to fuse with the infected cell, thereby forming a multinucleated cell and thus allowing the virus to travel through. Some of these ones, when the disease is so much, it forces the neighboring cells to fuse up, and in fusing up, the membranes will fuse together, and then there is a road, there is an open road for the pathogen inside the cell to move to the next cell. Equally, some pathogens also infect the immune cells, like we know about the GLAs, the CD4 cells. When HIV comes, the first attack is those. And then when they now attack and reduce the number, it tries to make them not recognize those cells. So some pathogens will attack the immune cells, and so can pass from one cell to another cell as the immune cell moves within and in between cells. We know that some of these immune cells will move. If you have a disease or if you have a problem within you, they will be moving around to control this. But some of these pathogens will attack them and reconfigure them and become part of the immune cell, so that as the immune cell is moving from one cell to another, they are transported by that same immune cell, who is supposed to, prior to destroy them, but they will not do that. Then, however, the following roles is in disallowing the pathogens to proliferate from one cell to another. This mechanism is what we might call the defense mechanism, that the cell also tries to not allow it to establish itself. Establishing physical and chemical barriers against invading pathogens. One cell can do that because this is going to proliferate. It will establish physical and chemical barriers. Such barriers are the membrane walls, tight junctions, which disallow the movement of pathogen between cells, and microbial peptides and enzymes that kill invading pathogens. We know, so, for example, like the lymph nodes and all that, they create barriers that disallow certain pathogens to move within our cells. I've had a case where somebody had cancer of the breast that has lasted for more than 7 years. But this could not pass through the lymph nodes, but through the breast. And that was removed successfully, and the person is still living. So another one, since the pathogens needed certain receptor proteins to attach and first invade new cells, such new cells can alter such receptor proteins, thereby preventing adherence and thus penetration into the cell. So these are another ways that external cells can protect itself from those pathogens which are already inside the cell from getting into it, and so disallowing proliferation to take place. Their cells may have defense mechanisms such as autophagy, in which the cell will engulf and degrade the protein. Certain genes in the cell, such as interferon stimulated genes, can be activated by the cell so that the viral infection can be blocked, degraded, and in some cases, apoptosis can be activated. This is another way. When that cell, that new cell notices that, oh, look, this cell is trying to give me a disease. It may block it from coming. It may also degrade it by engulfing it and destroying it. Or it may decide to, on its own, and say, okay, let me not have it and let me die. And it can die. Then cells can self-destruct, as we have said. The cell can duly use its immune system to survey and protect itself from infection. Such immune systems are like the natural killers we know, the cytotoxic T cells, which recognizes infected cells and destroy it. So in which case, if one cell is trying to send something to the next cell, and through those immune systems, they detect such a thing, they can force that, they can send their immune cells to go and recognize those cells and get it eliminated, in which case they protect themselves. So having seen what the new or uninfected cells can do to protect the cell, one would ask the question, outside the cell fighting for itself, protecting itself from these diseases, is it possible? We have other external ways in which we can externally help the cells to stop this disease from spreading within the cell and within the host, and thereby trying to now stop the disease from, in the first stage, even infecting anybody before we talk about controlling it in the population. And we said, yes, there are. One is enhancement of the immune system, which is most of the time we do by vaccines and other things. Enhancement of the immune system, which helps to fight off with this pathogen more effectively and efficiently. Two, by pharmacological means in which drugs are taken that tackles the pathogen at these early stages. Early stages like when they are trying to attach. Early stages are like when they have attached and have gotten themselves into the cell, will stop it from multiplying within the cell. And if you can stop it from multiplying within the cell, then there will be no proliferation. And if there are no proliferation, there will be no disease of the host. And you can do that by use of antibiotics, antiviral drugs, antifungal, and many others. The use of cell inhibitors that alter the cellular mechanism of the cell, and thus the interference in the pathogen development in the cell. Usually, when a particular disease is entering, by the time it has entered, it recognizes the structure of the cell. But when we now use some inhibitors that can change the cellular pattern of those cells, then the pathogen itself may be destroyed in the process because it will not find what it has recognized. Just like when a disease enters the body, the CD4 informs the bone marrow that, oh, look at this, produce a specific CD8 that can fight this disease. But if this specific disease is changed within that period, the CD8 may be released, but may not recognize the disease eventually because it's a different form. In the same way, the cell has entered, it recognizes the cell and the composition of the cell and the makeup of the cell. If the cellular mechanism is changed, the pathogen will be affected because it has not recognized that. Then we also have the prevention of the initial stage of infection by such methods as hand washing and distancing, like HIV, COVID-19 and others, or like the vector causative organisms like diseases like malaria and others. The application of this method highly inhibits the initial presence of the of course, we not present conditions for even pathogen penetration into the cell. So if you can use this, you can also block this. We are mentioning all this because in the model which we developed or which we are still developing, all this we are taking into consideration. And we are trying to get something that is, we're saying we don't even need to have a disease before we start talking about controlling the disease. Then we have again what we call the use of gene editing and biotechnology, in which the pathogen inside the cell can be genetically modified to become less infectious. This is another possible thing that can be done externally, not the cell doing it itself, but our medical people can do this. And if you can edit the gene of that disease, it can make it not to be very, very infectious. So from what we have seen and shown and now known, it means that the disease progression into and in the host can be obstructed or controlled at any of these levels, and thereby not allowing the pathogen to develop in the host in the first instance. This implies that it may be better and cost-effective to control the disease at these levels than allowing the disease to establish and spread in the population, and then control strategies sought to take out the disease in the population. And what are we asking? What we are asking is that we suggest that we shift our attention in biomedical modeling to these levels of infection, as it may be easier to handle or control the disease at these levels. Thanks. I didn't go ahead to produce the models because we are still working on it. But maybe over time, we're going to present the models that are here. I have already 3 PhD students working on this. But I want to show you that it is possible that we can still control the same epidemiological problems by not necessarily waiting for people to be infected before we can do that. But we can obstruct it, can stop the disease from even getting into the people in the first instance before we allow it to now escalate, and we talk about control within the population. Thank you, and God bless you. Hello?
Thank you, Professor Mbah, for your presentation and the insights into